All right, so hopefully you went and watched the fluid mosaic model video so that you could see the proteins floating around in that membrane in that video. Membrane proteins are really essential to how your cell works, how your cell communicates, how things work within your cell. So we're going to talk about those next. You have 5 different types of membrane proteins that we're going to talk about. You've got marker molecules, attachment proteins, transport proteins, receptor proteins, and enzymes. So, a lot of the effects of your plasma membrane, of your cell membrane, are determined by what membrane proteins are there, what combination you have. The ability of those proteins, to function properly is determined by their 3-dimensional shape. So, the thing about proteins, they are the workhorse of the body. They're the ones that make enzymes, they're the ones that make channels, they're the ones that do quite a bit. But when it comes down to it, if you want it to work the right way, it has to be the right shape. It's like your car. You wouldn't take the wheels of your car and put them on the roof. You wouldn't take the windows in your car and put them underneath the chassis. It just doesn't work that way. If you don't have the right things in the right place, it doesn't work. So we have 2 major types of proteins, okay? Integral or intrinsic and peripheral, extrinsic. What's your peripheral vision? The sides. So if you hear peripheral, that should give you a big hint as to where we're looking. So if you've got a cell membrane, okay, so here's my cell with my double layered membrane. Okay, so those are supposed to be my phospholipids. I'm not going to draw each one out. And you know what? I take it back. I'm going to blow this up so that we can see it a little bit better. So we've got our phospholipid bilayer. I know you're thrilled by watching me draw this, don't lie. The 2 types of proteins that we have are determined by how they sit in or on this membrane. If we're talking about an integral or an intrinsic protein, look at the first 2 letters, in, right? So this type of protein is actually going to be within the membrane. It's going to be stretching all the way across so that things can go in to my cell, or for that matter, things can go out of my cell. But it's transmembrane. Trans means across, so it goes across the membrane. Something to keep in mind, the tails still hate water, the heads still love water. So wherever this is in the area of these tails, okay, it's going to actually have to also be hydrophobic, just like those tails are. So right here, where it's right next to those tails, is actually hydrophobic. But the parts that are next to the heads, the parts up here, are actually going to have to be hydrophilic. Because just like the heads are right next to the water, this part of the protein is also right next to the water, okay? So you've got this, you've got this transmembrane protein that has different parts that kind of match up to where it's sitting. Now, peripheral, we said peripheral meant on the sides, right? So a peripheral protein would either be on the outside out here, or it would be on the inside in here. But it doesn't actually go through the membrane. That's why we say it's peripheral. Also, we call it extrinsic. Think exit. In other words, it's outside. It isn't going to be inside the cell. So I'm going to go back just for a second. That is a transmembrane protein. That big purple thing right there is a transmembrane protein. But if you look at this guy, he's only on one side of the cell. He's peripheral. Look at this guy. He's only on one side. He's on the inside of that cell membrane. So he's peripheral. We have 5 major classes of membrane proteins. We have marker molecules, attachment proteins, transport proteins, receptor proteins, and enzymes. I know this looks like a bear, but we're going to go through it all together. One of the ones that has kind of the most subsets of different kinds of proteins are the transport proteins. Okay? So, our transport proteins have channel proteins, carrier proteins, ATP-powered pumps. Channels can either be non-gated or gated. Gated can be voltage-gated or ligand-gated. Carrier proteins do exactly what they sound like. They carry, but the way that they carry varies. So you got uniporters, symporters, and antiporters. Now, the receptor proteins. Think of receptor as the word receiver. It actually gets something that it holds on to. So you've got receptors linked to channel proteins. Doesn't that sound familiar? Channel. I know it's science and it sucks that there's overlap, but there is. You also have receptors linked to G proteins. And then finally, we've got, well, I kind of already went through the big 5, but we've got the enzymes on the other end. So I am going to come back to this quite a bit, come back to this slide. You don't have to if you don't want to when you're following along, but I like to kind of show you where we're at. And we're going to start here. What's a marker do? Common sense. If I buy a house, they put markers up in my yard. Why do they do that? To show me where my yard is, right? So marker molecules are glycoproteins or glycolipids. We kind of already mentioned that. That glycoproteins and glycolipids can be used to identify your cells to other cells. Okay. Have you ever wondered how your body knows that you're infected with something? Your cells actually have proteins on their surface. I'm going to say this is a Ronica cell. Basically identifying your body cells as being body cells. When your immune system comes around and checks it out, it goes, oh yeah, that's a Ronica cell, that one's okay. That's what this is doing, okay? Now, let's say that I get sick and I've got the Ronica still on my cell, but this is a cell that has been infected with a virus. Okay? Something that's going to happen with this cell is it'll start displaying these really weird proteins on its surface that aren't normal. These types of things, when your body sees them and it doesn't go, oh, that's a Ronica, it goes, whoa, that's not Ronica. This is how your body literally says, okay, there's something wrong with that cell and we need to see about getting rid of it because it's not right. If you have a bacteria floating around in your body, they also have these marker molecules, except they say something completely different than your name. So they may say bacteria. And again, your body, your immune system checking these out goes, okay, that doesn't say Ronica, I need to kill this. So marker molecules can be integral, where they're transmembrane and they go across the membrane, or they can be peripheral. So immune cells use this a lot. Your immune cells are constantly scanning your body to see if there is something wrong with the cells that they see. It's constant, 24/7, 7 days a week, 365 days a year, your entire life. There are immune cells scanning your body. And they use these markers as a way to say, oh, yeah, that cell's okay, or okay, that cell doesn't look right. So that's marker molecules. Let's talk about attachment proteins now. Okay? And guess what they do with a name like that? Yeah, they attach to things. That's what they do. They can be integral proteins, and they may attach to intracellular molecules, in other words, something inside of your cell, or they may actually anchor to things outside of your cell. So an example is cadherins. Cadherins actually anchor a cell to a neighboring cell. Okay, that's what the protein is doing. It's actually attaching to another cell. Whereas integrins, like what you see up here, may attach to an extracellular molecule. It may attach to a collagen fiber, or it may attach to an elastic fiber, but it's not technically another cell. The integrins as well can also function in cellular communication. It can be a way of basically telling the cell you're in the right place because I'm anchored in the right place. So that is attachment proteins. So we've done marker molecules, we've done attachment proteins. Let's talk transport proteins. You know that in that table, transport proteins are kind of the one that has the most tiers. So let's talk about what a transport protein is. And basically, if I'm in the family transport protein, what do I show? If I am a transport protein, if that's what I'm labeled as, I exhibit these three characteristics, okay? One is specificity. So, specificity means if I bind to something, if I am transporting something, that is all I do. You're not going to find let's say a transport protein for sodium transporting sugar. It doesn't work that way. Literally, it usually binds to one type of molecule, and most of the time, that molecule is very, very specific. Unless we make kind of a man-made substitute that is pretty much the exact same thing, it only binds to one thing. So that's specificity. It only binds to the one thing that it transports, the end. 2, competition. So if you do have closely related substances that bind to the same binding site, the one with the higher or greater concentration or higher affinity, do you see that word right there? It basically means lovingness. I have a very high affinity for Jensen Ackles. I like him a lot, okay? Affinity means that you like that thing. So it's either a greater concentration or a higher affinity. That's the one that's more readily moved across the plasma membrane. So if we look at this example here, I've got a pink X and I've got a blue X. And let's say that they are closely related enough that this one transport protein will move either of them. Okay, so go with me with my story. Let's start with the concentration, okay, the greater concentration. Let's say, for the sake of argument, that I have 100,000 pink X's and I have, I'll go with 10 blue. Pink. Okay. Sheer numbers, just sheer numbers. The 100,000, that many is going to beat out 10. So who's going to move more? The pink is, because I've got 100,000 of those to move. The 10 blue can't compete with that. Now, affinity, the liking of. I like this more than I like that. When it comes to affinity, it usually means that one binds better to that channel than the other. So in this case, let's say that I have 100,000 pink, but I also have 100,000 blue. Okay? But let's say that blue has a higher affinity. In other words, the channel likes blue better than it likes pink. If the numbers are equal and it has a higher affinity for that blue, it's going to move blue because it likes it better. That's pretty straightforward. So there is competition, but there are ways to win that competition. Okay, so we've got specificity, we've got competition. Now, last one, saturation. Does everybody know what saturated means? Imagine taking a kitchen sponge, and I mean soaking it to the point that it is dripping. That's saturated. You got as much water in that sponge as it can handle, okay? So with saturation, the movement is limited by the number of transport proteins. The rate of movement will eventually plateau. It'll be going as fast as it can possibly go because the number of proteins are going at their maximum rate. So, if we're talking about time and rate, transport proteins will move really quickly at first, but eventually they do this, they plateau. Now, why does that happen? So let's talk about Walmart for a second. And I'm pretty sure this is universal. It doesn't matter what campus you're on. If you've been to a Walmart, you've seen this. So, Walmart. If I have 10 checkers at Walmart, so I have 10 checkers, okay? And they can move one client every 3 minutes. Okay, so each of these 10, every 3 minutes, they get done with one client and they check that client out. 3 minutes, new client, they can check them out. In 3 minutes, how many people can I check out with 10 checkers? 10. Because if each one of the 10 moves one client every 3 minutes, that's as fast as they can go. Does that make sense so far? I hope it does. So, if I have 5 people checking out at Walmart with my 10 checkers, nothing else has changed, I'm going to get done pretty quick, right? I can get 5 out in 3 minutes. I can get 6 out in 3 minutes. I can get 7 out in 3 minutes. I can get 10 out in 3 minutes. Now, what if I put 100,000 people checking out at Walmart all at the same time and I still have only 10 checkers? How many can I get out in 3 minutes? 10. I can make that number 1,000,000. It's not going to matter. If 10 checkers can only get 10 people out every 3 minutes, it doesn't matter how many people I put through. Eventually, there's a saturation point. There's a point where they're working as fast as they can, but it's not going to go any faster. That's what this is. That rate eventually plateaus. Those membrane proteins are moving stuff, they're transporting stuff as quickly as it possibly can, but now they're going as fast as they can. That's as fast as they can go. That's just the end of it. By making more people check out at Walmart, it's not going to make the checkers basically go any faster. If you can only get one client out every 3 minutes, that's the speed it's going to happen. So saturation is basically regarding the same thing. Movement is limited by the number of transport proteins, the number of checkers at Walmart, and the rate will eventually plateau. Again, if I can only do 10 people every 3 minutes, doesn't matter if I put 100,000 people in the line, it's not going to go any faster. Why? The number of proteins are all going at their maximum rate. The number of checkers at Walmart are all going as fast as they can. Just because I add more doesn't mean that it's going to go any faster. So if I fit into transport protein, if I fit into this category right here, I've got 3 very important characteristics. I exhibit specificity, I exhibit competition, and I exhibit saturation. Okay? So, anything below that umbrella, doesn't matter what it is, anything below that umbrella automatically fits into these 3 categories. If you don't see specificity, you're not going to be a transport protein. You can exhibit competition and saturation all you want. You don't have specificity, you don't fit. I show competition, great. I don't show specificity or saturation, I still don't fit. So, I'm going to go back a second. You're going to get sick of me doing this. Let's talk about this tier of my umbrella, okay? I've got channel proteins, carrier proteins, and ATP-powered pumps. Channel proteins form passageways through a plasma membrane. And just like our transmembrane protein we talked about slides before, it has hydrophobic and hydrophilic regions. So it's a doorway. Things can go into or out of my cell through this doorway. Okay? Everybody with me so far, I hope? Carrier proteins. They're a bit different because I'm talking about moving something bigger and bulkier. So if I come up to you with a hypodermic needle and I shove it in your arm, you're probably going to go, ow, but you're not going to bleed to death, right? However, if I take my fist and try to put that through your arm, probably wouldn't work real well, right? When we're moving things into and out of our cells, it's kind of the same thing. I can't just randomly make a hole, a huge hole in the side of my cell. So in this case, I have carrier proteins. Carrier proteins are kind of like revolving doors at a bank, okay? They move around and allow things into or out of, but if you pay attention to a revolving door, there's never a hole there. Whenever that door revolves, there's always a seal from where that person is standing. Most places that have revolving doors did it to either save on heat or save on air conditioning because they didn't want people doing what they do most of the time, which is they'll stand there with the door open. And as my mother would say, are you trying to air condition the whole neighborhood? What are you doing? Close the door, right? So with carrier proteins, we talk about a change in shape. ATP-powered pumps. They also move things across the membrane. The difference is when they do it, they use ATP to do it. The other 2 don't. The other 2 just move things, and you'll see why in a little bit. So, we've talked about these in general. Now we're going to come over here and start talking about the channel proteins. Remember, channels are doorways. We have two types of channel proteins, non-gated and gated. What does a gate do for you? It opens and it closes, right? That's what a gate does. I don't want anybody in my backyard. What do? You close the gate. Why? Because I don't want anybody back there. So it closes, nothing moves, right? At least that's the hope. Now, non-gated channel proteins are more like having an archway, okay? For the sake of this class, non-gated channels are always going to be open, okay? They're not going to have a way to close. Is that reality? No. But for simplicity's sake, we're going to say non-gated channels are always open. Now, if that's the case and they look like this, where there's no way to open or close them, that means that whatever's going through here goes through here. If I've got to move potassium and there is a potassium channel there that's a non-gated channel, potassium will just move. So we say that this is responsible for the permeability of the plasma membrane. Basically, if I've got 10,000 potassium channels, potassium has a way to get in and out of the cell really easily because I've got 10,000 of those channels. However, if I've got 3 calcium channels, calcium's going to have a bit of a harder time because there are only 3 archways for it to move through as opposed to 10,000. Okay? So again, non-gated, always open. They determine the permeability, how permeable, how much things move and what things move through our membrane. Now let's talk gated. Simple. A gate can be opened or it can be closed. That's what gated means. Instead of having just an open archway, now I actually have a doorway that I can open, or if I don't want it working, I close it. So we have 2 major types of gated channels, ligand gated and voltage gated. Okay? Ligand gated, you can kind of think of as having a key to a lock. So the ligand is going to be our key. Our channel has what we call a binding site. Some people call it an active site. But if the key isn't in the lock, so if you look here, there's no key here, the door is closed. However, if the ligand actually moves into that binding site, that active site, it opens the door for us, okay? It allows things in, it allows things out, depending on what we're moving. But for right now, what I want you to understand is the ligand is like a key. When it binds, the door opens, okay? Voltage gated. What do you think is in charge of opening the door? Voltage. Charge. So we've talked about ions. We talked about ions in the last chapter, right? Eraser. Okay. With a voltage-gated channel, you kind of got a little bit of a difference in how we open and close our channel. If you look, attached to the end of a chain here is this ball. So it's a ball and chain. And on that ball, I've got a specific charge. It's positive, right? So when we were talking about ions in chapter 2, I mentioned a really specific phrase. I said opposites attract, right? Look inside the cell here. What are the charges? They're all positive, right? So if they're the same, they don't attract very well when it comes to the negative positive thing. So what do? Well, if I'm the ball here and the inside of my cell is the same charge as I am, I'm going to shove myself into this channel to get away from all of these charges that are the same as me, effectively making me a door. Or if you want, you can call it a cork. Okay? If you've got a cork at the top of a bottle, is it going to spill? No. So having that ball and chain in place like that basically is the gate. It closes the door. Nothing can get past this guy. So it's a closed gate. However, if in the inside of my cell that charge changes, so again, I've got a positive on that ball and chain, right? If it becomes negative inside of my cell, that positive ball is going to go sub and move to get to the negatives. When it does that, it opens the door. So we call it voltage gated because literally that change from positive to negative inside the cell, that change in voltage, is what causes the door to open. It causes the gate to open. Go back again real quick. So that's this, and then these two. We've talked about all of that. Now, let's talk about carrier proteins. Okay? Carrier proteins move things from one side of the membrane to the other. And they do this by changing their shape. If you look at this example here, this is open in this direction. Oh, that's a terrible color. Hang on. Dun, da, da. Let's go with red. It's open in this direction in this picture. The little pink X comes in, and then it's open in this direction, kind of like a clamshell that went the other way, okay? So carrier proteins move things across the membrane, period. Uniporters. What does uni mean? Means one, right? Uniporters move one thing across the membrane. Period, end of story. Symporters. Sim means same. So symporters move two things, two ions, but they move them in the same direction. So if you look at this picture here, I've got A and B moving through the membrane, but both of them are going from outside to inside. So they're moving in the same direction. Then we have the antiporter. Anti means against, right? So again, we're moving two ions, but we're moving them in opposite directions. So if you can see here, A is going in, B is going out. I am moving two things, but they're moving in opposing directions. So those are the carrier proteins. So we've got the carrier protein. We talked about the uniporter, moves one thing, one direction. The symporter moves two things in the same direction. The antiporter moves two things in opposing directions. The sodium-potassium pump. Sodium-potassium pump has to have at least two binding sites, one for the thing that I'm moving and one for the ATP that I am going to break down and use. ATP is a fuel source. It's like putting gasoline in your car. You don't run your car off of the actual liquid gasoline. You burn that and then it powers your engine. So ATP, when you put it here, is actually broken into ADP. Remember, adenosine diphosphate with a phosphate, and then that charges up this channel. That charge that we just basically put into it is what allows us to move this molecule from one side of the membrane to the other. That charge is necessary for us to move things like that. Now, sorry guys. Whoops. That's the ATP powered pump. Now let's talk about receptor proteins. Okay, so we've done all of this over here. We've talked about all of this over here. Now we're going to be talking about receptor proteins. Receptor sounds like the word receiver, and that's exactly what this is. Oops. So, receptor proteins are proteins or glycoproteins in the plasma membrane that have this exposed receptor site. Okay? Remember back here when we were talking about channel proteins? I pointed out that there's that binding site right there with the key. Same kind of thing. Okay? Sorry. So you've got this receptor site on the outer surface of your cell, and it attaches to a specific chemical signal. So this may be, let's say, insulin, and this is an insulin receptor. This is growth hormone, and this is a growth hormone receptor. A lot of these receptor proteins are part of that remember, that means in between cells, cellular communication. They actually coordinate the activities of what's happening between one part of your body and another part of your body. The best example I can think to explain to you, remember we talked about a woman being in labor when we talked about the positive feedback loop. The chemical getting released from the brain was oxytocin. Okay, oxytocin is going to be the chemical signal. This receptor, the oxytocin receptor, would be in the uterus, so that when oxytocin gets there, this gets the signal that, oh, it's time for me to start contracting the uterus. So when I say it's a communication system, a way for the cells to work together, that's what I mean. So we know that we have two different types of receptor proteins. We have the receptors linked to channel proteins. We just went through that with our little blue triangle being the ligand and the green triangle being the ligand and the blue circle being the channel, right? This is the exact same thing, only this picture looks fancier. So we've got our ligand. In this case, it's acetylcholine. We've got this channel that has the binding site for it. And when acetylcholine is actually bound, like what you see in this picture, it causes the channel to open. Okay? Sorry, I dropped my pen. That is literally exactly what we just talked about. And yes, it is the same thing in both places, but when we talk about receptors, when we talk about receivers, okay, it's kind of its own thing because of this little dude right here. Receptors that are linked to G protein coupled receptors, okay, or G protein complexes. With channel proteins, when they bind, they actually change the shape of this thing that they're binding to. It goes from looking like this to looking like this, right? That's not what happens with G protein complexes, though. So, G protein complexes use something called a second messenger system. Basically, they get bound to, but then they send minions to do something in the cell. So I always think of Gru from Despicable Me, because Gru doesn't really do the work. He thinks up the ideas, but then he sends his minions to go and do what they do. So here's my chemical signal. Here's my chemical signal. Here's my G protein. Okay? So chemical signal, G protein. Here are my minions. You've got gamma in purple, beta in pink, and alpha in orange. When this is not bound to, notice nothing is binding here, These little minions stick next to Gru and they don't do anything. They're just sitting there going, you need us to do anything, you need us to do anything, you need us to do anything, and they're not doing anything yet. But when this chemical signal does bind, it's like a chain reaction where all of a sudden my G protein right here gives instructions to these little minions, gamma, beta, and alpha, that they need to go inside the cell and do something. So you'll notice here that alpha's heading for somewhere else, right? We call it a second messenger system because it's almost like a relay race. If you start a relay race, are you the one to finish it? Normally, no. Okay, so he binds to him, he gets the instructions, he gives the instructions to these guys, and these guys, my little minions, are the ones that go and do it. That's a G protein coupled receptor. Is there still something getting bound to? Is there still a receiver here? Yes, but the way that it works is completely different than the receptor on the channel protein. So, what can alpha do in the cell? Well, it can act as an intracellular chemical signal. Alpha itself can actually, the fact that it's floating around, cause stuff to happen. 2, it can actually open channels, but instead of from the outside like what we see up here, it floats from the inside and actually causes them to open from the inside of my cell. And then 3, the activation of enzymes associated with the plasma membrane. So I mentioned this before. I don't want my enzymes working unless I need them, but they're expensive. So if I don't need them working right that second, I also don't want them to just, be broken down into nothing. Again, that's like wasting a card. Let's not do that. So what do? We've got a key. In this case, in the case of number 3, basically what we're saying is that alpha subunit is the key to an enzyme. When this whole system gets activated, alpha goes and binds to that enzyme and boom, it now works. Now, when the G protein, when Gru loses this, alpha comes back, beta and gamma come back, and we get back to this kind of reset position. Finally, the last membrane protein we're going to talk about, and we've already talked about it twice, I believe. Enzymes. Enzymes can work on the outer surface of your cell. They can work on the inner surface of your cell. They can work in the plasma membrane in either direction. Of course, it depends on which proteins. There are some that are specific to the outside. There are others that are specific to the inside. It just kind of depends on which one you're talking about. Some of these enzymes are going to be active all the time. We need them all the time. They're going to be active all the time. Others, like I said before, are kind of like, if I need them, I'll turn them on. If I don't, then I don't. Okay, so these are the membrane proteins. Guys, membrane proteins are really important to how your body works, and we're going to discuss them a lot this semester. So it's really important you understand what these things are and how they work.